Blood type depends on the presence or absence of certain molecules, primarily proteins, on the surface of red blood cells. Cells are labeled as self by proteins on its surface. Cells lacking these self markers are recognized by the body's defense mechanism as foreign or non-self and attack. To the body's defense mechanisms, these proteins are antigens. These antigens identify the cell as foreign and mark it for destruction. One means is the antibodies produced by plasma cells target the antigen on the foreign cell's surface for destruction. Blood types are named by the antigen or the protein found on the surface of red blood cells. Type A has only the A antigen, type B has only the B antigen, type AB blood has both A and B, and type O has neither A or B on its surface. The table shows the different blood types, the antigens on the red blood cells, and the antibodies found in the plasma. So for blood type A, you see it has the A antigen on the red blood cell surface. Thus, it would have anti-B antibodies in the plasma. So blood types that they might receive in a transfusion would be either type A or type O, because recall that type O has neither antigen on its cell surface. Type B has B antigen on its surface, therefore it has anti-A antibodies in the plasma. So type B individuals could receive either a transfusion from a type B or a type O. Type AB has both A and B, therefore they have no antibodies in their plasma. And you can see on the blood types they are considered a universal recipient, because they can receive a blood transfusion from a type A, a type B, a type AB, or a type O. A type O individual, on the other hand, has both anti-A and anti-B antibodies. They can only receive a transfusion from a blood type A, a blood type O, so therefore they're considered the universal donor. Agglutination or clumping occurs when someone's antibodies contact a foreign cell. This response can also be used to determine blood type. If a wrong blood type is given, it can be damaging or fatal. Clumped cells that block blood vessels, they break open, releasing hemoglobin, which then clogs filtration in the kidneys, resulting in death. In blood tests, they can mix a drop of a person's blood with a solution containing anti-A or anti-B antibodies to see if this agglutination occurs. RH is another important factor to determining blood types. RH term comes from the term rhesus monkey, from where it was first discovered. Individuals who have RH antigens on their red blood cells are considered RH positive. Individuals who lack RH antibodies on their red blood cells are considered RH negative. So in this image, to test for type O blood type, recall that they have both anti-A and anti-B antibodies in their plasma. So when anti-A solution was added, you see no clumping occurs. Anti-B, no clumping occurs. When checking for anti-RH, you see that clumping did occur. So this was used to determine that this individual was O positive. On the other hand, looking at where they're testing for blood type A, under anti-A, clumping occurred. Anti-B, which is contained in type A individuals, no clumping occurred. And when anti-RH solution was added, clumping did not occur, which allowed this individual to be classified as blood type A negative, because no clumping occurred under the anti-B, designating this individual as blood type A, and also no clumping occurred under anti-RH, signifying this individual as RH negative. And this just shows the different diagrams a little bit closer of agglutination, where at the top there's no agglutination of red blood cells, but at the bottom is where these red blood cells agglutinate or clump together, which can be damaging or even fatal. This shows even a closer diagram of the link up where no agglutination occurred. The antigens that are found on the red blood cells themselves, antigen A in this case on the red blood cell, with antibody to B within the plasma, and at the bottom where a wrong blood type, antigen A and antibody to A, where they link up, causing this clumping to occur. An Rh negative person will not form anti-Rh antibodies unless he or she has been exposed to the Rh antigen, such as either during a transfusion or in the instance of having given birth to an Rh positive baby. An Rh negative person should only be given transfusions of Rh negative. If given an Rh positive, it will stimulate production of anti-Rh antibodies. Typically, this will not occur after the first transfusion because the body takes time to start making these anti-Rh antibodies. However, after a second transfusion of Rh positive blood, the antibodies in the recipient's plasma will react with the donor's blood, which can lead to death. If an Rh negative mother carrying an Rh positive child, typically maternal and fetal blood do not mix, although it can occur as a result of a miscarriage or a delivery in which some blood may mix. At that point, the mother would then produce the anti-Rh antibodies. Anti-Rh antibodies could then be passed to a fetus during a subsequent pregnancy. Anti-Rh antibodies, unlike red blood cells, can cross the placenta. Hemolytic disease, the newborn. Anti-Rh antibodies, as we just spoke, can develop in the mother and cross the placenta, destroying the Rh positive fetus's red blood cells, in which the baby may die or become very anemic. Of course, the Rh antigen is also determined from our genetics that we get from our mother and our father. So therefore it would be possible that the father's Rh antigen is being expressed in the fetus. Rogam disease has decreased dramatically in recent years because of Rogam. Rogam is a serum containing antibodies against the Rh antigen. It's given to an Rh negative mother to destroy any Rh positive fetal cells in her circulation that may have occurred, and thereby preventing her production of anti-Rh antibodies. This RhoGAM shot is given at approximately the 7th month of pregnancy and shortly after delivery of the baby if the infant is Rh positive. Rh antigens are thus prevented from being set in the memory of the mother's immune system. The injected antibodies disappear after a few months, therefore no antibodies linger to affect any fetus in a subsequent pregnancy. And so this imagery shows that an Rh positive man and an Rh negative woman can have an Rh positive child, baby, fetus. During the first pregnancy, some of the Rh positive blood of the fetus may enter the mother's circulation. After delivery, the mother begins to form the anti-Rh antibodies, and on a subsequent pregnancy with an Rh positive fetus, the anti-Rh antibodies can pass into the fetus's blood, causing its blood cells to burst. We'll move to blood clotting now and steps that occur once a blood vessel is cut. Well, the blood vessel will first constrict. Platelets are going to form a plug, sealing that leak. And chemical will attract more platelets to that site. Aspirin and ibuprofen prevent the formation of this chemical and therefore also inhibit clotting, such as in a heart attack. So that's why people a lot of times, if they're in the phase of a heart attack, will take an aspirin. And after these platelets begin to cling, a blood clot will form. The key events in clot formation are that clotting factors are released from the injured tissue and platelets. These clotting factors convert an inactive blood protein to prothrombin activator, which is produced by the liver. Prothrombin activator is going to convert another protein, a plasma protein called prothrombin, produced also by the liver, to thrombin, an active form. Thrombin is going to cause another plasma protein produced by the liver, termed fibrinogen, to form long strands of fibrin. And fibrin are the strands, or fibrin are strands that trap blood cells and form the clot. The clot is the barrier that prevents additional blood loss through the wound in the vessel. Pardon me. So going through this again, injured vessel in step 1, the blood vessel wall cells, the endothelial cells, and platelets are going to release clotting factors. Step 2, these clotting factors convert an inactive blood protein to prothrombin activator, which is going to activate prothrombin to thrombin. Thrombin will activate fibrinogen, which is going to activate fibrin to form the network, kind of like a spider web, to trap these platelets and forming this plug. When the wound is healed, there's another protein or enzyme called plasmin, which is formed from plasminogen, which will digest these fibrin strands of the clot. Plasmin is also produced in the liver. So once healing, plasmin formed from plasminogen will digest these fibrin strands. If even one of the clotting factors is lacking, the process can be slowed or completely blocked. Vitamin K is essential to synthesize prothrombin in the liver. Blood clotting does not occur without it, and there's two sources in which we get our vitamin K. Is diet and from the intestinal bacteria, so which can be killed if people take a lot of antibodies. So it's very important that even though we have to take antibodies sometimes for our bacterial infections, that overuse of antibodies can kill a very beneficial bacteria in our intestinal tract that produces vitamin K, which would be essential for blood clotting. Vitamin K is used rapidly by the body from both sources of diet. So vitamin K deficiency can occur in as few as 2 days. Hemophilia, because hemophilia is an X-linked recessive, it's more common in males. Symptoms seem to appear as a child when the child becomes more active. Excessive internal bleeding can damage nerves, or if joints can also lead to crippling. It's caused by a faulty gene involved in the production of clotting factors. There's 13 clotting factors identified, and they're numbered from 1 to 13, and they're just numbered in the order in which they were identified. Treatment involves restoration of the missing clotting factor for hemophilia, or could occur from a transfusion of fresh plasma. A concentrated clotting factor combines donations of several people. Textbook noted that each injection requires approximately 2,000 to 5,000 individual donors. A couple of clotting factors are now being manufactured using recombinant DNA. So this is an area in which genetic engineering has proven useful for human purposes. Formation of unnecessary blood clots can have immediate health consequences, as we know. Blood clots can disrupt blood flow and cause heart attacks and stroke. A clot that continues to circulate is referred to as an embolus. So if an embolus blocked flow to the heart, it would result in a myocardial infarction or heart attack, but the proper name is a myocardial infarction. Or if it went to the brain, it could result in a stroke. A clot that lodges in an unbroken blood vessel is referred to as a thrombus. Blood doping is the term used to describe the practice of boosting the number of red blood cells in the bloodstream in order to enhance athletic performance. Because red blood cells carry oxygen from the lungs to the muscles, a higher concentration in the blood can improve an athlete's aerobic or oxygen capacity and endurance. One means in which athletes may blood dope is that they withdraw blood from the body, which is going to prompt the body to produce more red blood cells. The blood is then going to be returned to the body. Athletes such as that swim, cycle, endurance, they need increased endurance. This method is prohibited in the Olympics, but it's very difficult to detect. Means in which they look for evidence of blood doping that they might test for would be to look for an increased number of red blood cells in the individual's blood. or the presence of the hormone erythropoietin produced by the kidneys, because recall that erythropoietin from the kidneys prompt red blood cell production. However, the test can only identify approximately 50% of those who have actually partaken in the blood doping.